So for this part of chapter 20, we're going to try to finish it in this recording. 20.2 is about prokaryotes, and of course we know what that is already, so we're just going to go over it really quickly. It includes bacteria and archaea, which are fully functioning, living, single-celled organisms. So the difference from remember we went over viruses before? The difference here is these guys are living, okay? They're fully functional and they're living. These guys don't really necessarily need something to host them. Today, we know that bacteria are plentiful in the air, water, and the soil. So the structure of prokaryotes is surrounded by a plasma membrane. We've already went over this a long time ago in Biology 103. And a cell wall situated outside of the membrane. So they have your plasma membrane, just like a eukaryotic cell, and then they have a cell wall on the outside of it that's really rigid, and it serves as like it's almost like it's bones, okay? The cell wall prevents the cell from bursting or collapsing. It's its structure, and it kind of keeps its shape. Some have an additional covering layer of polysaccharides called a glycolax, and it's really sticky. So if you'll remember, I'm sure you don't, but if you'll remember that saccharides are sugars. These guys are sugars. And there's two different types of these sugar layers or third layer. If it's well organized, it's a capsule. And if it's loosely organized, it ends up being like a slime layer, like a sticky outside layer. So it is like this. Helps it stick to stuff like your teeth, things like that. So my pictures here, when I downloaded this, kind of went over a little bit of text, but it's okay. So we already know this. So some move by means of flagella. A filament is inserted into a hook and anchored by a basal body. So this is, I didn't put that in there very well. That's what a flagella is. And we've already went over this. It's these guys right here. They have this basal body. They have a hook. And then they have this filament that kind of goes like this. And then when you turn this hook, when you twist it, it's like if you put a pipe filler in your hand. Is that what those things are called? I think anyway. But anyways, those little wire things with a fuzz on them. If you put one in between your fingers and you bent, like made a little hook, and you twisted it, it would literally go like a propeller. And so that's what we talked about. I think it was in chapter 4. That's what we called this. We called it our propeller. So many adhere to surfaces by means of fimbriae. These are little bitty like hair-like fibers extending from the surface. This gonorrhea, or I guess that says gonorrhea, attached to the host cell and cause, oh yeah, okay, there we go. That's what I knew. Those two things are caused by this bacteria. I'm not even going to try to pronounce it. And its bacterial cell has these tiny little fimbriae kind of hair, little attachment thing, or little guys that come out here and help attach to things, attach to the host cell. And this is really important. We already know this. They have a nucleus. I'm sorry. Ugh, look at me. They do not. That was really stupid. They do not have a nucleus. They have a nucleoid region. Because remember, these guys have no membrane bound organelles. So they just have this region in their cells, and they have their DNA that just kind of hangs out in the middle. They also have things called plasmids, which are rings of DNA. These help with antibiotic resistance, production of toxins, and they break down some chemicals. So sometimes little bacterial cells will share these little plasmids, and that's where the antibiotic resistance comes into. They're like, hey, I know about this drug. They're trying to get rid of us, Here's the code for it, or something like that. So the reproduction, I'm tired of seeing blue. The reproduction of prokaryotes, mitosis, we already know all this, the formation of the spindle apparatus. This does not occur in prokaryotes. They have a thing called binary fission. So if you think of the word fusion, what do you think of? Something that's fused together. You think of coming together, right? Well, this fission is the opposite. So it's going to, if you think about fusion coming together, then fission is going to pull apart. It's a single circular chromosome replicates, and then two copies separate as the cell enlarges and eventually leads into two cells. This only takes about 12 minutes. So eventually you'll have this guy that looks like this, and it's eventually going to pull apart in fission and make two. It's almost just like mitosis, but it's for the whole thing. They don't have a nucleus and things like that. They don't have to form a spindle or anything like that to pull their DNA apart in their nucleus, because they don't have one. Okay, in eukaryotes, genetic recombination occurs as a result of sexual reproduction. But they don't have that, okay? So they have to recombine a different way. So conjugation, two bacteria are temporarily linked together and pass DNA. So it's almost like they hold hands. And pass DNA. There's one type called transformation. This is where the cell just almost like picks up pieces of trash DNA that are secreted by others or released when one of the cells dies. So this is, I'm just going to, picking up trash. Then you have transduction. This is where bacteriophages carry portions of DNA from one to the other. So remember the last part, or the previous section, this is viruses that infect bacterial cells. So those viruses are carrying pieces of that DNA from one to another. So those are ways that they genetically, or they get different genetically, okay? Genetic recombination. That's how they do it. That's why eventually they're not all the same. The bacteria evolves over time, and these are the ways they do it. These three ways, okay? 1, 2, 3. So now we're going to talk about it. This was just generally talking about prokaryotes. There's different things, and prokaryotes is a huge umbrella, and one of the things under the umbrella is bacteria, okay? One of the main ones anyway. So we're going to get into bacteria in 20.3. So the characteristics of bacteria. Most are protected by a cell wall composed of peptidoglycan. And so if you go into biology like I did, you'll have to, in microbiology, I had to stain cells. So the picture that we're about to see, or that you see on the bottom, is actually what, I had to do that in my lab. It was pretty cool. And what we did was, you just stained a portion of the cell wall. And if it stained, it had peptidoglycan. If it didn't, it was the other one. So there's things called gram-positive and gram-negative. Gram-positive means they had the wall. They do have the peptidoglycan wall. And gram-negative means they had a really thin one. And then a second plasma membrane on top of that often blocks the antibiotics. So you can see the difference here. These are two different cells. It's pretty cool to do that. If we have time, I will try to order some things. I don't really have access to the lab money at North Jackson, but I'll try to order some things and let you guys stain some bacteria. I think we've already went over this, the shapes that a bacterial cell can be. The main ones we go over is the circle, the spiral, and the bacillus. It looks like a peel to me. My nose is bothering me. I think that was in chapter 4 as well. So something we might not have talked about in chapter 4 is the growth arrangement, okay? So a lot of bacteria is named or categorized by their shape and their growth arrangement. So staph means clusters. Strep means chains. And diplo means pairs. So here's staph right here. Here's strep, here's your chain, and here's your diplo. So a lot of the bacteria that I've heard of in labs and things like that are these two. You'll hear of Streptococcus. That tells me that it is in a chain and it's circular. Or Staphylococcus, that means it's in a big cluster. So you have your shape and you have how it grows together. So the metabolism of a bacterial cell, it can be many different things. They can use oxygen and then they don't have to use oxygen. There's ones that don't. So aerobic ones require oxygen to grow, okay? Need oxygen. I think about swimming when I hear of, that's what I always think of. I think about swimming because you need, I guess aerobic, I don't even know why I think about swimming, but I think about swimming and I think about aerobics and needing air when you do things like that. Like when you exercise. There's facultative anaerobes. That means there are both. Oh, that is a terrible color. They grow in both. And then there's things called obligate anaerobes that cannot grow in oxygen. So let me erase that swim here. So they need O2. Those can do both, and those can do no oxygen, okay? Some examples of your obligate anaerobes are botulism and tetanus. Bacteria that grows in your intestines or in deep puncture wounds, they can get in there. So autotrophic bacteria, we're getting close to somewhat being finished. We're on the tail end of it anyway. So photoautotrophs use photosynthesis. They use solar energy to reduce CO2 to organic compounds. So they same thing that plants do. Okay, that's where that prefix photo comes from. There are two types. 1, 2. The first one does not produce oxygen, like green sulfur bacteria. And the oxygenic one produces oxygen, like cyanobacteria. And we're going to look at cyanobacteria in a minute. Anoxygenic and oxygenic. There's two types of photoautotrophs. So you've got photoautotrophs and then you've got chemoautotrophs that use a thing called, they are chemosynthetic. They oxidize inorganic compounds, such as hydrogen gas, hydrogen sulfide, and ammonia. But regardless, this one uses the sun. This one uses the sun. And this one uses gases. But regardless, they both take CO2 to make food. Okay. One uses the sun, one uses gases. Chemo uses gases, photo uses the sun. Then you have heterotrophs, okay, some heterotrophs. So that means, we'll just go over them. Let me change this. Chemoheterotrophs obtain carbon and energy in the form of organic nutrients others produce. So if you remember that heterotrophs do not make their own food, okay? Autotrophs make their own food automatically. Heterotrophs have to get it from somewhere else. So example, they feed on tissues and fluids of the host. It's kind of gross. One type is called saprotrophs. They serve as decomposers that break down organic matter from dead organisms. So this happens when you pass roadkill. Saprotrophs are busy at work. It says chemoheterotrophs are used to make acetone, butter, cheese, silk, coffee, and some other antibiotics. It's kind of cool. Now we're going to talk about the relationships of these. Symbiotic relationships is when two different species are living together, and there's 3 types, okay? Species living together. The first one is mutualism. That should be pretty easy. Both benefit. Commensalism. Number 2, one benefits and the other one is unaffected. One usually modifies the environment, so the other benefits. It says obligate anaerobes live in intestines only because E. coli uses up the oxygen. So the E. coli uses up the oxygen so that obligate anaerobes have a place to live. And if E. coli wasn't there using it up, obligate anaerobes wouldn't be able to be there. And number 3, we've already talked about a parasitic relationship. One benefits and one is harmed. We talked about pathogens with viruses. So this is an important note. It says they aren't actually doing the harm. The toxins that they release are. An example they give you is tetanus, release of toxin that travels throughout the body. We talked about a pathogen not necessarily wanting to kill their host because they wanted somewhere to live, right? It's just a side effect. Endospores. I don't know if you've ever heard of endospores before, but they're dried seed. They're among the deadliest pathogens produced. The reason that they are come from some of the deadliest pathogens is because it's almost like putting something like laying dormant for as long as they want to. And they can just survive like all these kind of harsh environments and crazy temperatures and just nut stuff. They can live forever. And then they can just pop out and just start, killing things. It says anthrax. The anthrax endospore from 1,300 years ago can still cause a serious infection. This is botulism from canned food. Another important note is this is not means of reproduction, but just survival. I'm going to keep going. You guys can pause me if you need to. All right, so antibiotics. There are 2 types of antibiotics. One type of antibiotics inhibit protein synthesis, like your ribosomes, things like that. The next one inhibits cell wall biosynthesis. So some examples of these from the first one, the ones that inhibit protein synthesis, erythromycin, I know you've heard of that before, tetracyclines. The ones that inhibit cell wall biosynthesis, so that means these antibiotics kind of say like, stop, you're not producing any more cell wall, so those bacterial cells can't be made anymore, they can't reproduce or go through binary fission. Things that stop that is penicillin and ampicillin. So doctors will give you a certain antibiotic. It's also important to note that antibiotics, if you've never heard this before, don't treat viruses. It's important not to take a lot of antibiotics because you're allowing that your body to kind of get used to it. And if it is a bacterial cell, and you take them all the time, then you're just gonna get it more immune to it, and it's just gonna replicate and figure out a different way to get around it. So they're almost losing their effect because we take them all the time. But antibiotics do not treat viruses because viruses don't have a cell wall, and viruses don't have their own protein synthesis mechanisms, right? Oh, look at me. That's my next bullet. So it says heavily prescribed to treat infection, often when not needed. Bacteria is becoming increasingly resistant to antibiotics. They can even pass their resistance to others by, we've already talked about all three of these ways that they pass gene information. Staphylococcus aureus, which is something I used to mess with in the lab when I was in college, was only 3% resistant to penicillin when it came out. Now it's over 90% resistant because we overprescribe and we overtake. All right, we're almost done. A few more slides. The last thing we're going to talk about is cyanobacteria. So cyanobacteria is gram-negative with some unusual traits. It photosynthesizes, okay? Some people call it the blue-green algae, but it's not an algae. It's a prokaryote. It has a crazy color, and I'll show you in just a minute. You've seen it before. It has no means of locomotion. So how do you think it can survive without moving? Well, it uses the sun to eat, okay? This is cyanobacteria, probably making you dizzy. It can photosynthesize. It can also fix nitrogen gas, so no need to move for food. It also serves as food for some of our heterotrophs. It can be found in fresh marine waters, soils, hot springs, harsh environments, things like that. So phosphates can build up and drain into lakes and ponds, resulting in a bloom of this organism. So if we don't take care of we've probably seen this around in our water. What also happens is this layer of cyanobacteria covers the water, and light can't penetrate, so then the fish die, okay? I used to draw fish like this. Wait, hold on a minute. I used to draw fish like this all the time when I was little. The fish will die if you don't take care of this. That's what it looks like. I know you guys have seen it before. It almost looks like oil. It's different colors because it photosynthesizes. And that's it.